Hi and welcome to the 7.1 DNA structure and replication video. There's quite a few difficult concepts in here, so
This
if you
is
need
specification
if you want to
take a
stop
unit.
and have a read of it. Okay, so we're going to start with the Hershey and Chase investigation. Now, Hershey and Chase were the scientists that provided evidence that the DNA was the genetic material within cells that was passed on from parent to offspring. Within their investigations, viruses and bacteria were used, and it's worth noting that a phage is a virus that can infect a bacterium. That terminology comes up quite a lot, comes up in this diagram here and when you are researching. It might also be useful to know that viruses have a protein coat containing sulfur. We should know by now that amino acids can contain sulfur, and that the genetic material contains phosphorus. So you should know that within a nucleotide you have phosphorus, and also there isn't any sulfur in the genetic material and no phosphorus in the protein coat, which is why it was those two elements that they decided to label. Okay, so in experiment 1, which is this one here in the green box, the phage is grown here in a radioactive sulfur solution. Now, what that does is it makes the outside protein coat radioactive. What then happens is they mix the phage with bacteria, and we can see here, and we know this now, but Hershey and Chase didn't know this, that the virus is injecting the DNA into the bacteria. Now, it has to do that because the bacteria is the host cell, and viruses can't reproduce without a host cell. So we can see here that there's no radioactivity within the cell because this part of the virus doesn't enter the cell, this part of the virus leaves, and this is where the radioactivity is. Now, once all of that has happened, we centrifuge the solution. So this is all happening within the solution. We centrifuge the solution. And what then happens when we centrifuge it is we spin it round at really, really high speed. And at the bottom here, we end up with the bacterial cells. So within this, we call it a pellet. We end up with bacterial cells. And above there is a liquid called a supernatant. And within the supernatant is the radioactivity. So we can therefore deduce that whatever was injected into the bacteria that is passed on to the virus offspring is the hereditary genetic material, and this protein coat, which is in the supernatant and not inside the bacteria, is not then passed on to the viral offspring. In experiment 2, you can see that same thing, but the DNA within the virus is labeled. That DNA is then injected into the bacteria, and within the bacteria you have the radioactivity. So the phage is allowed to reproduce and reproduce. The bacteria and the phages are mixed within a solution. And the reproduction occurs. We centrifuge the solution, so round and round and round and round, and then we have our pellet and the supernatant. You can see now that the radioactivity is within the pellet. So remember, the bacteria are in the pellet. So within the pellet are the bacteria, and inside the bacteria is the radioactivity, or the radioactive phosphorus. And then Hershey and Chase deduced that DNA is the genetic material that is passed on to the offspring. And that's just kind of summarized here. So that's the Hershey and Chase investigation. Okay, so the structure of DNA. So we're going to look at how we now know what the structure of DNA is and who the scientists were that found out what the structure of DNA was. So in 1952, a female scientist called Rosalind Franklin worked with an assistant called Maurice Wilkins, and they produced photo 51, which is this photo here. And I'm going to be completely honest. I don't really understand how they made these assumptions from the photo. They're obviously way cleverer than me, but this is photo 51, and it was produced using X-ray diffraction. Again, I don't know a lot about that. However, it's called photo 51 because it was the 51st X-ray diffraction picture that they produced. Now, from this picture, what they learned was that this center cross indicated that the DNA was a helix. And the cross also helps them to see the steepness of the angle of the helix. I don't know how. And then the distance between these small lines within that cross proved that each of the turns of the helix were 3.4 nanometers apart. Again, I don't know how they worked that out, but they did. So we need to know that Rosalind Franklin produced this photo 51 with the Morris Wilkins. In 1962, Watson and Crick built the first model of DNA, and they used Franklin's work, but they didn't ask her permission. She was maybe dead by then. I can't remember when she died. Watson and Crick suggested the sugar phosphate backbone model, which we now know to be true. And they suggested that the nitrogenous bases were in the middle of the helix with the sugar phosphate backbone on the outside. They were also the first to suggest that the 4 bases were either purine bases or pyrimidine bases. Now, purine bases are 2-ring bases, pyrimidine bases are single-ring bases, and the purines are A and G, and the pyrimidines are T and C. So a purine always has to be paired up with a pyrimidine. The model also led to the hypothesis that DNA replication was semi-conservative, which you should already know about. We also need to know that it's worth noting as well. Watson and Crick won the no, this should be a capital letter. Sorry, Watson and Crick won the Nobel Prize for their model of DNA using Franklin's work, but she wasn't credited because she was already dead when they won it. Okay, nucleosomes. So here you can see that you've got, this is your DNA helix, so the double helix here, and then here you've got the helix being wrapped around some proteins. We call those proteins histones. So each of these is a nucleosome. So a histone wrapped in DNA is referred to as a nucleosome. So nucleosomes are made up of DNA and histones. Histones are the proteins that DNA is wrapped around. This helps with the supercoiling of the DNA. So when we talk about the supercoiling, we talk about this, but also you can see here that the histones are arranged in a specific pattern, and then they are packaged into the chromosome. And we call all of this packaging the supercoiling in order to make the chromosomes. And the reason that we have to supercoil DNA is so that the DNA can fit into the nuclei of the cells, otherwise it would be too big to fit in. Okay, onto DNA replication. Now this can seem quite complicated, but I'm going to try and make it as simple as possible. So key terminology. We need to know that there is a leading strand and a lagging strand within DNA replication. You've done DNA replication in SL. It's actually far more complicated than you are led to believe. Now on this diagram, you can see that your DNA helix has unwound. We've got one strand here, this strand with all of the, this strand here with the green bases. So this strand is referred to as the leading strand. And that's because within the leading strand, DNA replication can occur from the 5' to 3' in a 5' to 3' direction, and it's continuous. It goes all the way along the strand, and we make a new strand as we learn to SL. On the lagging strand, however, the DNA, the new strand of, we can't replicate in a 5' to 3' direction, and therefore we need a slightly different process in order to produce the complementary strand of DNA. We'll go into more detail on that soon. What we have to do is we have to make these small fragments of DNA called Okazaki fragments. So these Okazaki fragments are produced in a 5' to 3' direction, and then they're all joined together to make your big long strand of DNA. You might also see the term replication fork referred to. That's just where the DNA is split. So this here is your replication fork, because replication is occurring where the DNA is kind of forked. Okay, in the exam questions, they tend to ask you about the enzymes involved in DNA replication as opposed to you just talking about it as a process. And the reason being because when you list the enzymes in the right order and describe their function, you actually are talking about the process in order. So the first enzyme we need to know about is DNA helicase. So DNA helicase unwinds the DNA, just as you've learnt previously, nothing new. You've also, helicase is here, this blue triangle. You've also got this one here, which is called DNA topoisomerase, or sometimes referred to as gyrase. Now, what this does is as you unwind your DNA strand, this relieves the strain, because if you unwind something really quickly, what happens is it kind of the strain in it bunches up. So this relieves the strain. I think I've spelled relieve wrong there, sorry. It relieves the strain on the unwinding DNA. Enzyme number 2. Enzyme number 3 is RNA primase. Now, it's DNA primase there. Can't remember. I think okay. DNA or RNA primase. You might need to look that one up. I'll put it in the description which one's the correct one. Sorry. What the DNA primase does is it adds on an RNA primer. Now, the RNA primer is just a single base or a short sequence, and what that does on the lagging strand is that kind of says to the other bases, you can start adding on to this strand from here. So the primer adds on, and then all of the other nucleotides add on after that primer. So you've got a primer there, and this is also a primer here. So you've got nucleotides that have added on after that. And that happens all along that strand. So primer binds, and then you've got all of the other nucleotides that bind after it. And once they've bound after that, it produces the Okazaki fragment. Now you've got DNA polymerase 3, that adds DNA nucleotides to the growing Okazaki fragment. So DNA polymerase 3. Don't worry too much about this DNA polymerase. We'll talk about that later. But DNA polymerase 3 is the enzyme that adds on these extra nucleotides once the primer has been added. DNA polymerase 1, once all of the Okazaki fragments have been produced, the DNA polymerase 1 removes the RNA primer, because we don't want an RNA nucleotide within our DNA strand. So it removes the RNA primer and it fills the sites with the corresponding DNA nucleotide. And then finally, once you've got all of these Okazaki fragments made, the DNA ligase seals the nicks between the Okazaki fragments in order to make one continuous DNA strand. So the primer attaches, all of the nucleotides attach after that, producing an Okazaki fragment. DNA polymerase removes the primer, replaces with the DNA nucleotide, and then DNA ligase links all of the Okazaki fragments together.